The Aggregate Variants per Gene tool aggregates the VCF files in the specified folder for each gene and mutation type. After dividing the sample into the control group and case group, press the Aggregate button to run the aggregation. Check only the missense and frameshift variants for the variant type and press the Filter button. In the population filter, we try to extract the mutations that do not exist in control group but always appear in case group. There are 238 genes found. Let's see them by pressing the Export Table button. The results are stored in Aggregate per Gene folder. The TSV file is a table of aggregate products. There are columns for gene names, number of occurrences of each sample, mean frequency in the control group. Those in the case group follow. This is a list of genes having mutations in the case group, and the number of frameshift and missense variants for each gene. Return to the Tools screen and press the Export Bed button. You find the bed files for each sample and the summary of the case group. You can visualize the bed files by importing it as a region list. Please use the option to add CHR to the prefix. Import the bed files of each sample in the same way. These scores are the quality of the VCF. You can drag and drop the imported region list onto the genome view to display each region as a track. In the Genome tab in the lower panel, search for the ABCC6 gene. In the Chromosome tab, drag around the found gene to display the area in Genome view. Zoom in around ABCC6. In this case, you can see that the different mutations are found in each mutant sample. Expanding further, we can see that there are 2 mutations in one exon.